Welcome to our video lecture on Introduction to Cells, our very first lecture video in IB Bio. I hope you're so excited. Real quick first, remember that there is totally optional lecture outline on Canvas if you want to annotate that online or print and write on the paper. Take notes how you want to take notes, but I highly recommend that you do take notes because remember that all our quizzes this unit will be open note. So take some notes. Our lecture also is going to start with this essential understanding. IB is always going to give us an essential understanding for all of our lessons. This is just the big idea that we should come away with at the end of our lesson. And our essential understanding for today is that, according to cell theory, and we're going to talk about what that is, living organisms are composed of cells. We're going to talk a lot about cells today. Good. All right. So before we get going too far, I do want us to think about the objectives. What even do we want to learn in today's lesson? So we're going to be able to state the tenets of cell theory. There are three. You need to memorize them. We're going to list examples of exceptions to cell theory. There are three of those that we're going to talk about. There are more than three. We're going to talk about three. We're going to talk about functions of life, what makes living things living and non-living things not living. We're going to describe the emergent properties of organisms, which is super cool because it's kind of like a combination of biology and philosophy. So big brain ideas. We're going to define stem cells, talk about different kinds of stem cells, and I'm going to talk about how we can use those stem cells in therapies, different ways that we can treat diseases and injuries with stem cells. We're going to talk about the ethics of doing that too. We're going to outline some of the theories of the origins of life and the origins of cells, and then we're going to talk about one of my most favorite theories in all of biology, endosymbiosis. All right, so before we do that, let's talk about some of the vocab that you probably already know. These are words that you probably heard back in intro bio. If you want to pause the video and take a look at these, if you want to look any of them up before we get going, you can. We are going to talk about all of these in today's lesson. But noodle on these, activate some of the knowledge that you already have, and get ready, get set. Here we go. So cell theory, there are three statements, three tenets of cell theory that you need to have memorized. So the first two sound pretty similar to each other. Living organisms are composed of cells. And then this one, same idea, just coming from a different angle. Cells are the basic building blocks of all living things. So memorize these two. Living organisms are composed of cells. Cells are the basic building blocks of all living things. Number three, cells arise from pre-existing cells. They don't pop up out of nothing. They have to come from something. And that something is another cell. So in general, our cells are pretty small. They have a nucleus or a nucleoid where the DNA is. They've got a cell membrane that kind of holds them. And then they've got some other organelles that look like super cool things. So this is our generalized cell. Living things are composed of cells. Most of our cells are small. They've got a space for their DNA, either a nucleus or a nucleoid. They've got some organelles. They've got a cell membrane, and they're pretty small, except our exceptions to the cell theory. And that's what this guy is here. So take a look at this guy, long and stringy. So this is muscle fiber. This is striated muscle. Sometimes we call it bone or skeletal muscle. This is basically like your quadricep or your bicep. And the cell that I've outlined here is too big to fit on the microscope slide. It keeps going off of the image. That's really, 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 really big. So these guys are unlike other cells because they're giant. And then also you might notice all of these little spots in here. These are nuclei. What do you mean, nuclei? Yes, my friends, those are all nucleuses. And so one of the other reasons that we consider striated muscle to be an exception to the cell theory is that it's multi-nucleate. It has multiple nuclei. It has a lot of nuclei. That's weird and different, and therefore this is an exception to cell theory. Another exception to cell theory are these guys. This is algae. And what's kind of crazy is this algae, that guy right there, being held by this person's hand, that's a single cell. Also, this guy right here, single cell. This guy right here, that's one cell, just one cell. And so this algae is known as giant algae because even though like clearly there's the person's hand, it's not that giant. Those are giant cells. Each and every one of these guys is a cell. That's amazing and weird. Therefore, it's an exception to cell theory. One more exception that we're going to talk about is this guy. This is part of a fungus. Fungi do not have roots. They do not have roots. They have what we call hyphae, which kind of look like roots, but they're not. They're not roots at all. They don't function the same way as roots. The great majority of a fungus actually is this underground hyphae system. And what's super cool, kind of like our muscle cells, this is a single cell that has some multiple nuclei. And so we call this aseptate hyphae. They're pretty big, crazy long filament cells. And they are also multinucleate. They have multiple nuclei. Super crazy cool. So I mentioned that this is what we see. What we don't see, this is like the underground part of the fungus. What we do see pop up are these fruiting bodies. Those are the reproductive structures of the fungus. And we can also see those reproductive features right there. So here are the fruiting bodies of bread mold. Good. All right, enough of that. So mold is alive and we are alive and rocks are not. So I want you to noodle on what makes us and bread mold alive, but rocks not. Hopefully you came up with some of these things. So here are the functions of life. And what's really cool about these functions of life is that single-celled organisms, unicellular organisms can carry out all of these functions in their single cell. And then those of us who are multicellular, we can carry out those functions using groups of cells. So this organism, the single-celled organism, is called a paramecium. It is one of IB's most favorite unicellular organisms. And you can see that it has a nucleus like all cells, well, not bacteria cells, all eukaryotic cells ought to have. It has a cell membrane, but what's super cool is that it's got lots of cilia. These cilia are used for movement. It's got some cilia here and what we call the oral groove. And the cilia here help to move food down that oral groove. And so that's how paramecium carry out nutrition. How they get food into their cells is with cilia and these oral grooves. Once the food gets to the bottom of the gullet, the oral groove, that food is going to be put into a special kind of vesicle called a food vacuole. Enzymes are going to get added to that food vacuole and digestion, metabolism is going to occur. So metabolism is both digestion, and then once we have digestion done, we take the little pieces and parts of our food, we put them back together in new ways. So metabolism is both breaking things down and building new things from our food. So that metabolism is going to occur. Sometimes these particles that we ingest and metabolize are used to grow. And so this paramecium cell can get bigger and bigger and bigger. Oh gosh, hold on, sorry. We can also respond. And the paramecium can respond to the environment. So if things change, if it gets too salty or too warm or whatever, the cell can act in a different way to respond to those changes in the environment. The cell can also get rid of waste products. So when we metabolize our food, sometimes we have gunk that we need to get out. Paramecium can do that just like we can. One of my favorite things that paramecium do is through homeostasis, when it's trying to keep its water balanced. So Paramecium live in ponds, and ponds have more, they're freshwater, so there's more stuff like salt and sugar and protein. More stuff is inside the cell than is outside the cell. And so water through osmosis is going to move into the cell. So osmosis of water, that water is going to cross the cell membrane of the paramecium and it's going to fill up the cell. And you can imagine that if this happens too much, we're going to have a giant cell that could just like burst. But the paramecium has this amazing organelle right here, the one that looks like a little bit like a starfish, called a contractile vacuole. And the vacuole is going to pull the water into itself until this guy gets bigger and bigger and bigger, and then it just squirts it all out. It's the most amazing little squirty movement that you have ever seen. So again, the water moves in. This guy gets bigger, 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 and then it just squirts all that water out. So water balance in the paramecium, helped by contractile vacuoles in us. Our kidneys do that job, along with our brains and wanting to drink water and some other things that we do. And then last but not least, our final function of living things, reproduction. Of course, we can reproduce our cells through mitosis. We can also reproduce entire organisms. We're going to watch a couple videos of Paramecium doing super cool things. So this one is a Paramecium eating. So look for the cilia, the little hair-like structures that move the Paramecium around, but also move the food down the oral groove into the gullet. Look for those food vacuoles being formed, and enjoy the background music, which came with the video. It's not me. So here we've got some Paramecium, and you can see those bubbles of food vacuoles rolling around. You can see the cilia flapping the food right there down into the oral groove. Notice that cilia on the outside of the cell too, helping him grow, helping him move. Those beautiful food vacuoles full of particles from the pond. All right, we're going to go ahead and skip to the next video where we're going to watch Paramecium divide. All right, so here we're going to go through Paramecium reproduction. We have music again. Again, it's the music that came with the video. It's a very different genre. Get ready, get set. Here we go. And we're going to skip ahead through this one a little bit and turn that music down a little more. So here you see Paramecium that started division. You can see like the new cell membrane starting to form across the middle. We're going to skip ahead a little bit. You can see how he's starting to separate more, the indentations between the two cells. We'll skip up a little bit more. And you can see that it's just barely hanging on. And then finally, they're going to wiggle themselves apart. And there they go. Two baby Paramecium, just like that. All right. So sometimes IB wants you to label the body of a Paramecium. So we're going to go ahead and practice labeling the body of a Paramecium. Oh gosh, that was not what I meant to do. Okay, so here we've got that nucleus. Of course, we've got some cell membrane. We have all these beautiful cilia that are involved in movement. We've got some food vacuoles. Those food vacuoles are produced when food particles are swept down the oral groove into the gullet by more cilia. So these cilia are also involved in movement, but now they're moving the food particles down the gullet into these vesicles, food vacuoles, so that those food particles can be used for energy, for metabolism. And then our favorite, the contractile vacuole. So because this paramecium lives in a pond, the fresh water in the pond is going to, via osmosis, diffuse into the cell. The cell is going to get bigger and bigger and bigger. That's bad because the cell could burst. And so that water is pulled into the contractile vacuole, which gets bigger and bigger and bigger. And then it's just going to squirt all that water right back into the pond. And then it happens all over again. So super cool parts of the paramecium. Make sure that you can label these chunks. Good. All right, let's talk a little bit about multicellular organisms like us as opposed to unicellular organisms like the paramecium. So this idea of emergent properties is basically that the whole is greater than the sum of the parts. Us as a whole organism can do a whole lot more than a pile of our cells could do. And so the interactions between cells to make tissues, the interactions amongst tissues to make an organ, the interactions amongst organs to have the system, the interaction of all our different organ systems to make us the organism function. This is amazing. And what we can do as a whole human is way more than all the things that the cells could do by themselves put together, if that makes any sense. So us, the whole, is much greater than the sum of our cells. We can do more than our cells could do individually. So super cool. Us humans, we have 220 different kinds of specialized cells. These cells in our bodies arise through differentiation and gene expression. So all the cells in our body, they all have all the same DNA. They have all the same genes, unless you're like a red blood cell, in which case you don't have any DNA because your nucleus dissolves. But that's a story for another day. Most of our cells have all the same DNA, have all the same genes. We just turn on and turn off those genes in different ways in different kinds of cells. So I could start with this single cell, which I could refer to as a stem cell. And the stem cell could go through mitosis, and I could have one cell, one daughter cell from that stem cell, in which we turn on or express genes that are specific to neurons. And then this cell is going to grow into a nerve cell. I could take a different daughter cell from that same stem cell, and in it, I could express or turn on epithelial cells, and I could have the inside of a small intestine, or the inside of my nose, or the inside of my esophagus. And so we can turn on, turn off different kinds of genes in order to get different kinds of cells from our stem cells. Stem cells are amazing, my friends. So we're going to talk about different potentials. So different potentials of different kinds of stem cells. So potent means like powerful. What kind of potential is there for these different cells? I'm sure lots of you have been told that you have so much potential. Well, do you have as much potential as these totipotent stem cells? Actually, you probably do. So we're going to look at fertilization here. So I've got sperm cell, we've got egg cell. The combination of these forms a zygote, a single cell, which is going to go through mitosis to form what we call a morula. This is a solid ball of cells. All of these cells are exactly like all of the other cells. And then a super cool thing starts to happen. The morula turns into a blastocyst, which is a hollow ball of cells. We go from solid, and those cells like migrate out to the outside of the ball. And so now I have a hollow ball of cells. Here's just like fluid on the inside. And then we get this inner mass of cells that starts to collect on one side of the blastocyst. We're going to have this thing called gastrulation occur. There's this little divot. And then at that point, now our cells go from being totipotent stem cells. They could totally turn into any kind of cell, a placenta cell, an amniotic sac cell, or an embryo heart cell. So now these inner mast cells can only turn into organs. These guys have lost some of their potential. These guys cannot make placenta cells. They have become, instead of totipotent, now they're only pluripotent. These guys are going to go through some more differentiation depending on like that little divot that happens in the gastrula, and we're going to end up with some cells that can only be digestive tissue. Like all your plans are only in the digestive system. You might be small intestine or stomach or large intestine, but you can't be a brain cell. We have other cells that are going to differentiate into nervous tissue, so brain and spinal cord. We have some that are going to be cardiovascular tissue, so a heart cell. These heart cells are unipotent. They can only be heart cells. When these guys go through mitosis, they turn into more heart cells. They cannot turn into pancreas cells unless we do some stuff, and I'll talk about that in a couple more slides. Some of our adult cells are multipotent, like bone marrow. Bone marrow is multipotent. It can turn into red blood cells or white blood cells, lymphocytes, leukocytes. But a lot of our adult cells are unipotent. Heart cells can only become more heart cells. So again, we have these different levels of stem cells. Some can turn into anything. These morula cells are totipotent. Our embryonic stem cells are pluripotent. And then we've got adult cells that are multi or unipotent. They have the potential to become different things at different levels. So one of the ways we can use our stem cells to help cure people of diseases is in this disease that we call Stargardt's disease. The retina is the chunk of nerves in the back of your eyeball. So this is what actually sends signals to your brain so that your brain knows what you're seeing. And a healthy retina looks like this picture on the left. This chunk is called the fovea. And then around the fovea is the macula. And this is the densest region of nerve cells. So this is where we have like our keenest, most acute sense of vision. This is where we see the best. You can see over here, someone with Stargardt's disease, that's gone. So the fovea, the macula have begun to atrophy. They're shrinking. They are degenerating. This person is going blind. What we can do is we can actually take some stem cells and we can insert them into the retina. We can put stem cells from a donor and we can put them into the retina. The chemical markers of the retina are going to induce these stem cells to differentiate into retina cells, functioning retina cells that can fill that hole and restore the vision of this person with Stargardt's disease. Another disease that we can treat with stem cells is Parkinson's disease. Parkinson's disease is a disorder of the nervous system, results in lack of control of movement. And so people move very slowly, they react very slowly, they fall down very easily, but then their bodies also move on their own. And so you can imagine that if your arms and legs are moving, tremors all the time, it's exhausting and terrible. It's a terrible disease to live with. And so what causes Parkinson's is we have these nerve cells in the brain that are not producing the dopamine that is necessary to control movement. We can take some stem cells, we can plug them into the brain. The chemical signals around these stem cells will induce the nerve cells to differentiate into nerve cells that do produce the appropriate amounts of dopamine, and that allows this person to recover. Their symptoms are lessened because those dopamine neurotransmitters can do their jobs. So lots of amazing things can be done with stem cells. Just a quick overview of lots of other things that we can do with stem cells. We can take these pluripotent stem cells. So these are from an embryo. And we can use these cells to turn into bone marrow cells, which we can use to treat leukemia, bone marrow transplants. Anyone who has had serious chemotherapy is lacking some bone marrow, and so we can actually replace their dead bone marrow with some new stem cells to replace that bone marrow. We can use stem cells to differentiate into skin cells for burn victims. Islet cells are in the pancreas, so people with type 1 diabetes, we can treat their diabetes with stem cells that can differentiate into cells that produce insulin. People with heart disease, we can use stem cells to make new heart muscle. We talked about Stargardt's disease and replacing retina cells. We talked about Parkinson's and replacing nerve cells with stem cells. So lots of amazing things that we can do. The challenge of our using stem cells is that often we have the ethical dilemma of the fact that a lot of our stem cells come from embryos. So we have to take that inner cell mass from a blastocyst. We have to harvest those embryonic stem cells. And when we take those out, that blastocyst can no longer grow into a human. So when we harvest these cells, we're also preventing this human from developing. We can do a lot of things with those embryonic stem cells. We can also find some stem cells in adults. It's a little bit harder because adults don't have as many stem cells. It can be very painful to the adult who's donating the stem cells when we extract them. There just aren't a lot of them. It's pretty expensive to find them. But we could pull retina cells out of an eyeball, perhaps from a cadaver instead of a very recently formed cadaver. And we could pull out some of those retina stem cells, and those could become the cells that we need to treat Stargardt's disease. Bone marrow, we can of course extract that and use that in transplants. We can pull the lining out of the insides of our noses. Those can be used in transplants. We can pull some stem cells out of adult brains and use those. We can also, and this one we'll talk about a little bit more in the picture that's under this one, we can actually take some cells and we can induce them to become stem cells, and then we have those cells that we need to treat the disorder. Sorry, I don't know what I just did. Clicked too many times. So talking about these different ways that we can reprogram cells, so reprogramming cells. So one way is somatic cell nuclear transfer. What I'm going to do what we do in this process is we take an adult somatic cell. Somatic means body. So somatic cell, it's a body cell, nuclear transfer. I'm going to transfer the nucleus of a body cell. So I take an adult cell, like a pancreas cell, and I extract its nucleus. I take an egg and I take its nucleus out, and then I plop in the nucleus that I stole from the pancreas cell. This is now going to go through mitosis. We're going to end up with the morula, with the blastula, with the gastrula. I'm going to have a ball of embryonic stem cells, just like I would in a regular old embryo. Advantages, I have a beautiful clone of this adult cell, and I can use those stem cells to do the work of that adult cell. They work a lot like embryonic stem cells. The disadvantage is I'm basically making an embryo and then I'm destroying the embryo to harvest the cells. So the ethics come into play again. We also have nuclear reprogramming, and this is amazing. I can take some adult somatic cells, again, these are just regular old body cells, and I can add reprogramming factors. So some chemicals, some molecules, some complex molecules that treat these cells and make them go backwards in time, and they're turned back into pluripotent cells. These stem cells are a lot like adult donor cells. So like if I pulled bone marrow out of an adult, these guys are a lot like those bone marrow cells, but I didn't have to pull bone marrow. I could just take like blood. The problem is those reprogramming factors are oncogenic retroviruses and transgenes. Oncogenic means cancer forming. And so it's kind of amazing that we can treat these cells to turn them into stem cells, but we also tend to turn them into cancer cells. And if someone is sick, the last thing that they need is to get more sick. So lots of ethical issues surrounding the use of stem cells. And so policy comes into play. There's, of course, a pretty tight connection between science and society. Lots of policies are involved. Lots of policies have been written that regulate how we can use stem cells. So here in the USA, different presidents, different parties in charge have certainly led to different policies on stem cell research. So in 2001, there was no federal funding allowed for any stem cell research done on new stem cell lines. So if you already had a pile of stem cells in your lab, you could keep using those, but you couldn't harvest any new stem cells. That changed with the new president. We took that restriction away. Any embryos that were created for reproductive purposes, like in vitro fertilization, any embryos that were left over and donated by parents, those could be used for stem cell research. And you might be thinking, well, why would parents donate their embryos? Well, as of November 2021, which is the most recent data I could find, here in the United States alone, just in the United States, we have 620,000 embryos frozen, waiting, just waiting. 620,000 frozen embryos. So parents could choose, they don't have to, but they could choose to donate those embryos for research. That changed in 2019. There was no longer any federal funding allowed for research involving fetal tissue. And also anything involving stem cells required this National Institutes of Health review that was time consuming and very expensive. So basically, stem cell research got shut down then. That's changed recently. That decision, that 2019 decision, was reversed. And then it also allowed, this 2021 decision also allowed for the use of stem cells that were donated by people who had elective abortions. We could use those stem cells in research. So lots of changes here in the United States over time. And depending on your personal ethics involving stem cell research, consider how you vote so that you can support those ethics that you hold. In other countries, there are different, well, sometimes similar, sometimes different policies. So in Great Britain, oh, geez, Louise, you can harvest, this is humic embryonic stem cells, so HES, human embryonic stem cells. If the research improves knowledge of embryology disease or disease treatment, I don't know who gets to decide that research is improving that knowledge, but hey, it's a good goal. In South Africa, if an embryo is fewer than 14 days old, you can use them for stem cell research, but you can't make an embryo to harvest it before 14 days. In Brazil, if the embryo has been frozen for at least 3 years, you can use them in research. In China, they're pretty open, but there is no human cloning allowed, and we're going to talk about that on the next slide because, oh my gosh, crazy things happened in China. And then in Germany, we can only use imported stem cells. Apparently, German stem cells are not used in Germany, which is kind of weird. And they have to be pre-existing stem cell lines. They cannot be any that are harvested recently. All right, so I mentioned China and some of the crazy stuff that's gone down. So this is a few years ago now, but still talked about a lot in science. So this scientist decided that he really wanted to cure HIV. What a great goal. And so what he did was terrible. And it did land him in jail. He got fired from his job. He went to jail for a little while, not long enough, if you ask me, but that's my personal opinion, not necessarily the right opinion. So what he did was he used CRISPR-Cas9, which is a way for us to edit genes. And he invited couples who couldn't have a baby because the dads were HIV positive. So he took HIV positive dads, he took their sperm, he rinsed the virus away from the sperm, he used the sperm to fertilize eggs from the women in these couples. And then he had embryos. In these embryos, he used CRISPR-Cas9 to edit some genes that would make these humans resistant to HIV, which is, again, a great goal. But CRISPR-Cas9 has not been around for that long. It has not been around for that long. We don't know long-term effects. We don't know side effects. We don't know lots of things yet. But this guy decided to go ahead and try it on humans. And so in China, there are twin girls. This article is old, remains pregnant. There was another baby girl born. So 3 kiddos in China who their genes were edited and they might be resistant to HIV. But how do we even test that? Let's not test that. He did go to jail. He was released from jail in April of 2022, so just recently. All right. So some people would say that's amazing that we can make people resistant to HIV, and other people would be like, whoa, that's too much. So some scientists would argue that we need to slow down, and others are like, it's just the science. Let's figure out the science, and society can decide what to do with it. So this scientist from Stanford, you can read his words. We need to slow down as a global civilization. My question to us, who comprises global civilization? Does like every single person on the whole entire planet get to vote on this? Do just the people who can read get to vote on this? Do just the scientists who understand exactly what's going on, all the details of it, do they get to decide? Do our politicians get to decide? And so noodle on the ethical dilemmas involved in using this biotechnology on people. Like, who gets to decide to slow down or speed up or use the technology or don't use the technology? There's not an easy answer to that, by the way. All right, so we're going to flip away from ethics for a little bit and talk about how did we even get these cells. We talked about cells come from pre-existing cells, but at some point there had to be the first cell that all the other cells came from. So how did we get to that first cell? Well, it wasn't through spontaneous generation. So a couple hundred years ago, we didn't know all that much science. It was kind of sad. We had this idea, society had this idea of spontaneous generation, which was that cells could arise from not cells. So not part of the cell theory. And the reason that we had this idea was that people would notice like piles of manure, poo, like you'd have a pile of poo, and if you left it outside, a day later, there might be some worms growing on the poo. And then a couple days after that, there'd be all these baby flies that were buzzing away from the poo. And you were like, ha ha, flies come from poo. That was wrong. And this guy, Louis Pasteur, was the one who helped us debunk spontaneous generation. So Louis Pasteur, and you've probably heard of pasteurization, which is the process of heating stuff up to kill the germs in it. Well, that's exactly what Louis Pasteur did with these super cool pieces of glassware called swan neck flasks. So Pasteur took his swan neck flasks, and he had some broth that probably had some microorganisms in it, but he didn't have a microscope, so he didn't know about those microorganisms, but they were probably there. But he heated up to kill off all the stuff in the broth. And the vapors could escape, but when he left this guy sitting, any bacteria or fungus spores that might be in the air could fall down, but they got here. Bacteria don't have wings. They cannot fly up and away. So all the bacteria, fungus, protists, they got trapped right there, which meant that nothing could get to the broth and grow. And for years, nothing was growing. Oh, by the way, his broth, he used like dog urine. Yay! Yum! I can't imagine the smell of heating up dog urine. Anyhow, so for years, the dog urine, nothing grew in it, and it was amazing until the flask got tilted a little bit. And so the broth came into contact with all the stuff that got stuck here. And when he tilted his flask back up, mere days later, it was full of gunk. Because those cells were able to get to the food and reproduce to make more cells. So that third statement of cell theory, cells come from other cells. Thank you, Pasteur. That's why we know that. Um, but again, somehow, some way, there had to be that first cell, and we have lots of gaps in our knowledge understanding that still, but we're making progress. And one of the progresses, whoa, that's bad grammar. This guy Stanley Miller did an investigation in which he took some of the gases that we think existed on early Earth, and he took some water, which was kind of like the ocean, and he pumped that gas into a glass bulb where he had lots of electricity set up, and there was basically lightning. He zapped the primitive atmosphere, that ammonia, that methane, that hydrogen gas, with lightning. And so stuff happened because of all that energy being added, and he cooled off the gases. They turned into a liquid, and they dripped down in, and they dripped with the water. And then he found that from water and ammonia and methane and hydrogen, with that big zap of energy, some amino acids could be formed. Amino acids are the building blocks of proteins. Did he make a cell? Definitely not. Did he even make a whole protein or DNA? Nope. But my friends, a whole amino acid just from like zapping ammonia and methane, that's amazing. And so this is our first little bit of understanding into how did life arise here on Earth. There's also lots of evidence that organic compounds came here on objects from space, not like space ships, but asteroids, meteors, meteorites. I don't remember which one is the one that lands on Earth. Meteorites, I think. Regardless, rocks from space that made it all the way to the surface of the Earth may have also brought us some super cool organic compounds, which helped to speed up that process of the evolution of life here on Earth. One more idea, one of my most favorite theories in all of biology, endosymbiosis. Endo means inside, sym means together, bio means life. So these are living things together inside each other. So this idea was put forth by Lynn Margulis, a super amazing evolutionary biologist. And the idea is what happened here. We have a big ancestral host cell. It's a prokaryote. There's no nucleus. For some weird mutation-y reasons, the cell membrane started to fold in a little bit and formed like a little pocket around the DNA. And so we had an early nucleus forming. And then the cell decided, you know what? I'm going to eat this bacteria. This bacteria that is aerobic, it's really good at making ATP. And so the cell engulfed it in a membrane-bound vesicle, but instead of digesting this aerobic bacteria, the host cell decided, well, didn't decide. They don't decide things. They just happen. The host cell didn't digest it. Instead, a symbiotic living together relationship developed in which this host cell provided nutrients to the aerobic bacteria, and the bacteria made ATP that was used by the host cell. Over lots and lots and lots of time, this aerobic bacteria became what we call today the mitochondria, powerhouse of the cell. But wait, there's more. If you were super cool and you were a plant cell, well, a cell that was going to become a plant cell, then what you did was you also gobbled up, ingested a cyanobacterium, which is a bacteria that can do photosynthesis. And again, you used your membrane, you built a bubble around the it's not a chloroplast yet, it's a bacteria. And over lots and lots and lots of time, we have chloroplasts, and that's amazing. So we think that bacteria evolved into chloroplasts. Mitochondria evolved from bacteria. How do we know this? What evidence do we have for this idea besides it just being a really interesting idea? Well, first of all, and this mnemonic device, by the way, comes from BioNinja. BioNinja is one of my most favorite websites. So Mad Doctor is your mnemonic device to remember these pieces of evidence for evolution of mitochondria and chloroplasts through endosymbiosis. First of all, our chloroplasts and mitochondria have double membranes. Like literally they had their own membrane and then they had the other one that got formed when that vesicle was put around it. Double membranes. Our chloroplasts and mitochondria are susceptible to antibiotics. Only bacteria can be killed by antibiotics, except also mitochondria and chloroplasts can be killed with antibiotics like penicillin. Crazy sauce. They reproduce, mitochondria and chloroplasts reproduce by binary fission, just like bacteria. Mitochondria and chloroplasts have their own DNA. There's like little pieces of DNA in our chloroplasts and our mitochondria. And what's cool is that DNA is naked. It doesn't have any histone proteins, and it's also circular, which is just like bacteria DNA, not like the DNA that's in our nucleus. Finally, chloroplasts and ribosomes No, chloroplasts and mitochondria have their very own ribosomes. Remember that ribosomes are where we make proteins. Mitochondria and chloroplasts have their own ribosomes. They make their own proteins. And what's super cool about the ribosomes in our chloroplasts and our mitochondria is that they're smaller in size. They're the 70S size, which is the same as bacteria's ribosomes. The ribosomes that we have floating around in the rest of our cell are 80S ribosomes. They're just a little bit bigger. How cool is that? Chloroplasts and mitochondria have their own DNA and their own ribosomes. Oh my goodness, that's amazing. All right, we're going to review the objectives, and then, my friends, you are done with your very first lecture, and I know that it was long, but now we get to go to lab and have fun when you get to class. So hopefully by now you are able to state those 3 statements of cell theory. Cells come from other cells. All living things are made of cells. Cells are the building blocks of cells. Oh gosh, cells are the building blocks of living things. They don't build cells. All right. We talked about some of the exceptions to cell theory, that striated muscle. We talked about giant algae. We talked about aseptate hyphae of fungi. Functions of life, nutrition and metabolism and reproduction and excretion and growth, response, and metabolism. No, homeostasis. That's one I forgot. Emergent properties. Thus, the whole is greater than the sum of the parts. Work pretty cool like that. We talked about different kinds of stem cells, the totipotent, pluripotent, multipotent, unipotent kinds of stem cells. We talked about some different ways that we can treat disorders and diseases with stem cells, like Stargardt's disease. We talked about origins of life and cells and endosymbiosis. And my friends, good job.